Hi, I am Mrs. Sloan, and this video is all about the nervous system. I'm going to make myself a little bit smaller. For my honors bio students, this is chapter 17. Okay, so we're going to take this whole chapter and break it into two videos, all right? So let's start off, first of all, with the divisions of the nervous system in our introduction, in our notes. And by the way, if you're new to my notes, down in the descriptor of the video, you will see a link to the group shared notes my students use, and that contains a link to this entire presentation if you would like to have that. So it's two columns. Column one is a scaffolding. I'll help you fill it in. Column two, I encourage my students to put in pictures so they can use it during reviews. So the division. The central nervous system is basically your brain and your spinal cord. You can see it right here in pink. So central is brain and spinal cord, and peripheral nervous system is all the nerves that are attached to the brain and the spinal cord. So all nerves. Now, basically, you have sensory neurons and you have motor neurons. So sensory neurons go towards the central nervous system and motor neurons go away. What connects the sensory and motor neurons are interneurons. Now, sometimes you can see here, this is a cell body. It has its nucleus. In sensory neurons, it's along this pathway right here, where in motor neurons, can you see that the cell body is part of the spinal cord? And we'll talk a little bit more about that later. For right now, let's have sensory neurons are headed towards the CNN. Motor neurons are headed away from the central nervous system. And where are they going to? They're going to either a muscle or they are going to a gland. Those are your only two effectors in your entire body. Think about it. This is how you respond to people when you talk to them, with a muscle or a gland. Okay? And then interneurons are found in between them, and I'll show you a picture. So typical neuron structure. Let's see, do I need to give you notes? I'm sorry, let me give you the notes for that. Interneurons are found in the central nervous system, CNS, between the sensory and motor neurons. So neuron structure. So dendrites are the part that receive the information, and then you have a cell body. In this case, with a motor neuron, the cell body is located right here next to the dendrites. But if it was a sensory neuron, you might see your cell body over here. Okay, so it's not always here. And then this long part here, this long part here is the axon. And so the information comes in via the dendrite and then travels along the axon and then leaves the neuron here at the axon terminal. And I'm going to pre-explain this. It's going to be an electrical signal along the length of the neuron, and then it's a chemical signal, which we call neurotransmitter, down here at the synaptic knobs. So on your neuron structure, dendrites receive a chemical message and usually send it through the cell body. The cell body contains a nucleus for processing, and then an axon takes the resulting message away, axons away, ultimately passing then a chemical message to the next neuron, or an effector at the synapse, or an effector at a synapse. Now, a synapse is just where two neurons meet, or if it's a neuromuscular junction, it's where a neuron meets a muscle. So the dendrites are going to receive a chemical message. This is going to be transferred into an electrical message, and then another chemical message comes out here at the end of the axon. All right, now surrounding the long axon in motor neurons, you're going to see this myelin sheath right here, this insulating layer. So on your notes, myelin sheath, insulation layer generated by what's called neuroglial cells surrounding these long axons. Now, you have little gaps in there called the nodes of Ranvier, so gaps in the layer. And the purpose is to insulate this neuron, just like wires. You know, we cover them with plastic, right? Okay, so insulates, it protects, and it speeds up conduction along the neuron. Now, I'm going to talk to you about how that electrical stimulus crosses along a neuron, and then it'll make more sense there. So we'll just put a pin in that for now, but it does speed up conduction along those neurons. All right, we got the basics, but let's review it a little bit. So this is a sensory neuron. So let's say it's coming from your skin, or it could be coming from your eye, right? It could be coming from your tongue, right? But here, this one's coming from your skin, and it's moving along. These would be the dendrites, the receptor, moving along the length of the axon. Here's the cell body. And then this sensory neuron right here would then synapse with an inner neuron in your brain or in your spinal cord. Okay. Now going away from your central nervous system would be a motor neuron. So this cell body and these dendrites would be inside your central nervous system, and it's going to conduct the information going out of your central nervous system towards your effectors. Your effectors are either going to be a muscle or a gland. So for, we already talked about that. It's in our notes. And then in between the sensory neuron and the motor neuron would be an interneuron. So this dendrite right here is receiving. It could be receiving information from a sensory neuron, right? And then passing that information on to a motor neuron. So putting it all together, let's say you're walking around your house barefoot and you step on a tack. So here's a tack. So here's the dendrite of the sensory neuron receiving information, and it's headed in, and this would be in your central nervous system. So this sensory neuron synapses here, its axon with the receiving dendrites of this interneuron. It's going to pass the information along its axon to a dendrite receiving of a motor neuron out to its axons, which will go into a muscle or to a gland. In this case, you'd probably pull your foot away. All right, so just think dendrites receive, it goes along the length of the axon and sends away on an axon. And where you have them connected together, that would be a synapse. All right. So now let's talk about that transmission. And I mentioned this earlier. Along the length of a neuron, it will be an electrical stimulus, okay, that passes an electrical message. And then here at the synapse between two different neurons, it will be a chemical message. And then again, an electrical message. So let's start with the nerve impulse and an action potential along the length of a neuron. That's what we're going to start with, that electrical message. Okay, and the first thing I need to do is I need to remind you of something we learned a long time ago, and that was the sodium-potassium pump. All right. Now, if you recall, the sodium-potassium pump is where you spend 30% of all the ATP you make from cellular respiration. And basically what happens is you have 3 sodium ions, and thanks to the ATP, you will expel 3 sodium ions to the outside of your neuron. And then once they have released the sodium, then 2 potassiums will bind and they will get released to the inside of the neuron. So it's 3 sodiums out, 2 potassiums in. 3 sodiums out, 2 potassiums in. Now I want you to think about that just for a minute. Do they have the same charge? Yes. Sodium and potassium are both positively charged. But where are we putting more positive charges? We're putting more positive charges to the outside than we are to the inside. So already we're creating a differential, more positives outside than inside. And when we talk about a neuron at rest, this is a neuron not firing, okay? Not firing yet. It can fire, okay, but it is not firing yet. And that is referred to as the resting potential. So let's look at what contributes overall to the resting potential, because the outside of a neuron, of a resting neuron is positive and the inside is negative. Why? It's a factor of 3 things. 1, I already talked to you about, that was that sodium-potassium pump, which pumps 3 positives out, 3 sodiums out, and 2 potassiums in. Let's look at those. In this picture right here, the sodiums are this blue color and the potassiums are this what looks like green color. So you can see there's a lot more blues on the outside and a lot more greens on the inside. The second factor that contributes to the resting potential of positive outside, negative inside, because basically we're making a charged battery here, is that inside the neurons there are negatively charged ions and proteins. So already more positives out than in, and on top of that, we have negative ions on the inside. And then the third factor is that the potassium gates are not that great of gates. And what happens is some of this potassium tends to leak back out to the outside. So it loses even more of its positiveness to the outside. So let's put this in our notes. On the nerve impulse, the resting potential, you are not firing, and it's when a neuron does not have an electrical message traveling along its length. The charges, the inside is negative and the outside is positive. Inside negative, outside is positive due to sodium-potassium pump, which puts 3 sodium ions out and 2 potassium ions to the inside. Overall, more positives out than inside the neuron. Number 2 was those negatively charged ions or proteins on the inside of the neuron. And number 3 is the potassium tends to leak back outside. Now, why is that important? Because we have a differential. Do you remember when we were learning cellular respiration and photosynthesis and how if we had more of something on one side of the membrane, like remember we talked about hydrogen ions, concentrating them on one side of the membrane of the mitochondria and the chloroplast. We had a difference in concentration, charge, and pH. Do you remember that? And we could use that because when they came back to the other side, we could use that for work and we could synthesize ATP, right? We are creating another differential now. And this is a difference in charge. And we can use that to do work, and that will be our electrical message that we're going to pass. That electrical message is called an action potential. So there's resting potential I'm showing you now, and then we're going to show how we can use that in our action potential. Now, in its most basic form, remember we said in the resting potential, the outside is positive and the inside is negative. Why? More sodium's out than potassium's in, right? Negatively charged ions on the inside, and some of this potassium tends to leak out. That we already learned. An action potential opens up the sodium gates. Now, sodium is going to want to go from where it's highly concentrated, right? It's going to want to diffuse. It's going to want to go to where it's less concentrated. So there's more sodium outside than inside. So it's going to want to come inside the neuron. In addition, you know that opposites attract. So if the inside it's negatively charged, those positive charges are going to be drawn to the negative charge. So sodium is going to come in, and at that point, these positives racing in are going to flip the charges. And the inside now overall is going to be positive. And in that one spot where the sodiums came in, then it's going to be negative in that one spot. This is called depolarization. We've depolarized it. It was, and now we've made the inside positive. Okay, now our quick fix, because this is going to happen along the length of the neuron, our quick fix to restore it to its resting potential is then the potassium gates are going to open, and potassium is going to go from where it's more concentrated to where it's less concentrated, okay? And that will restore so that the outside, again, is positive and the inside is negative. But if this keeps happening, if steps 2 and 3 keep happening because the neuron keeps firing, pretty soon you're not going to have any sodium on the outside. And these gates work one way, okay? They can only go from out to in, and these gates work one way, bringing potassium in to out. You are going to run out of sodium potassium. So your long-term fix is going to be your sodium-potassium pump, who's going to put 3 sodiums out and 2 potassiums back in. All right, so your action potential is an impulse, and it's going to start at the dendrites and work its way towards the axon. Okay, so we're starting here at the dendrites, and we're going to work through the axon. So look right here where it says action potential. Remember what we said that the action potential was, right? That was sodium coming in. And I'll talk to you about why these gates even open up in a minute. So just put a pin in it for right now. Just believe me, at this point, sodium gates are opening, all right? And so sodium's going to come from the outside to the inside. And the beauty of this is when this sodium comes in, it's going to trigger so that the neighboring sodium gates open. And so more sodium will come in, which then triggers its neighboring gates and so forth along the whole length of this neuron. It's kind of like dominoes. If you had a line of dominoes and you're going brrr along the length. And so this action potential is going to fire along the length of this neuron. Now, right behind the action potential is going to be your quick fix, right? This is when potassium's going to come from the inside and go back outside so that it evens up. It's like a do-si-do. Oh yeah, sodium, you came in, well, potassium's going to go back out. So as quickly as you're going and sodium's coming in, you're going and potassium's going back out, and it's fixing it, okay? And it's making it positive on the outside, and then it's returning it to its negative charge on the inside. And this will happen along the length of the neuron. In fact, it does such a great job, it will hyperpolarize it and make it super negative on the inside. Why is that important? It keeps the action potential going in one direction. So on your notes for action potential, an impulse starting at the dendrites and ending at the axon in the synapse. It is a chemical message is received at the dendrites, and I'll be talking more about that later, but that chemical message received at the dendrites begins opening these sodium gates. And we'll talk about that part later, but it begins opening these sodium gates. As sodium gates open, that's the word you need in your notes, as sodium gates open and sodium comes rushing into the neuron due to facilitated diffusion. Remember, diffusion is high to low, and when you use a protein to do that, it's facilitating that process due to facilitated diffusion. Okay, inside becomes less negative and may reach a threshold that triggers an action potential. Okay, which we're assuming is taking place here is this action potential. And it starts opening all the neighboring sodium gates, The inside becomes temporarily positive. Which triggers the next neighboring sodium gate. Okay, which triggers the next neighboring sodium gate and to open and so forth, like dominoes falling. The reversal of charge along the length, this reversal going along the length of this neuron, okay, is called the action potential. It is called an impulse or the action potential. And it transmits a message from one end to the other. This domino effect of reversal of charge, that action potential, a chemical message is received here, which triggers this domino effect, which then can release a neurotransmitter or a chemical on the other end, right? The repolarization takes place, repolarization of the resting potential. The short term is these potassium gates open, and I'm going to just go back here so you see that. The short term or the quick fix is that the potassium gates open and potassium goes out and takes sodium's place, counteracting the reversal, restoring it so it's positive again on the outside. The long-term fix is the sodium-potassium pump, putting sodium in its correct spot outside and potassium on the inside. So what have we done? We've shown that electrical change and restoration that occurs along that neuron to get a message from point A to point B, from the dendrite to the axon. All right, and we already looked at that. All right, so now let's look at the change in charge that we just talked about, okay? This stimulus right here, if it is enough of a stimulus, which we're going to talk about in the synapse, then it'll cross the threshold, which will trigger the sodium gates to start to open. When that happens, that depolarization takes the inside, remember the resting potential, the inside is negatively charged. It's got to get it less negative, less negative, less negative, less negative. Boom, we've crossed the threshold. The light is green. Then all of a sudden, all this sodium comes rushing in. So now the inside has become what? Positively charged. Why? Because all the sodium came in. Okay, that is your action potential. Now it is quickly restored, okay, and repolarized by what? The potassium gates, okay, the potassium gates open. So potassium goes from the inside to the outside and when it does that, right, it's taking its positive charge away and going out, then it restores it. In fact, so much so that the inside becomes hyperpolarized, becomes very, very negative, and this keeps it flowing in one direction, and eventually it diffuses away, and you're back to that resting state right here. Okay. All right. So that is the electrical stimulus. Now we're going to talk about the electrical impulse that goes along the length of the neuron. Now we're going to talk about the chemical message at the synapse. I want to remind you what the synapse is. It's a junction between two neurons. A junction between two neurons. Now, that's what you call a synapse. A neuromuscular junction is when a neuron is synapsing with a muscle, neuromuscular junction. Now, let's look right here. Here's an action potential that's triggered along this neuron. This is the synapse. If it's excitatory, it's going to continue that impulse along. It could be an inhibitory message. It could be saying stop. So we'll talk about that too. So here it is again. Here's a neuron where it synapses with the dendrites of another neuron, right? This is the axon, and it's connecting with the dendrites of a second neuron. That is the synapse. So if we blow that up, take a look. This would be the second neuron, the receiving or postsynaptic neuron. And this would be like a motor neuron. Look at the different colors here in red and blue. So here, the ones that are on the hair, some of them, the blue ones, are inhibitory message, trying to prevent this neuron from firing. These red ones are excitatory, trying to promote this neuron to fire. So a single neuron could have 10,000 synapses on them, some being excitatory, some being inhibitory. So the excitatory one has got to win. And by the way, those closest to the axonal hillock, where the axon starts, they're a little more powerful than the ones farther away from the axon, and whether it's going to fire or not fire. So on your synapse defined it's a junction between two neurons, can be 1,000 to 10,000 synapses per neuron. The presynaptic neuron, right, this is the one here that would be the presynaptic neuron, is sending the chemical message. The postsynaptic neuron here, which would be this guy right here, the postsynaptic neuron is receiving the chemical message. And then the synaptic cleft, there's a little itty bitty space, and I'll show you that in another picture. It's a small space between the neurons, All right, and this is what it looks like in reality. Okay, this is the cell body of a motor neuron, and these are all the little axonal terminals or synaptic knobs, all with a synapse right here, trying to control this motor neuron. All right, so let's look at the basics of it, all right? So this would be your presynaptic neuron here in yellow, your postsynaptic neuron down here in blue. And in its simplest form, at the end of those axonal bulbs, remember we said it's a chemical message, so it's stored in little vesicles, this chemical message, and that chemical messenger is called a neurotransmitter, okay? What will happen is, due to the action potential, this vesicle containing the neurotransmitter will move down and basically do exocytosis right here at the synapse and release its neurotransmitter into this space called the synaptic cleft. Now, you have receptors on the postsynaptic neuron, and this chemical, this ligand, will bind, okay? And what it can do, if it's excitatory, it will open up gates that allow sodium to come in and try to depolarize the neuron if it reaches the threshold, okay? If it reaches the threshold. Now, let me show you in a little more detail. Let's level it up a little bit, okay? So this is your presynaptic neuron. This is your synaptic cleft, your space. Here's your postsynaptic neuron. Okay, so here it comes. Let's go through it in the numbers. An action potential is traveling down the length of the neuron. What this will trigger is a new kind of gate to open, and that is a calcium gate. Calcium is concentrated outside the neuron. Calcium will go from a higher concentration to a lower concentration. The presence of this calcium causes these synaptic vesicles to migrate to the end of our postsynaptic neuron and release its neurotransmitter into the cleft. Okay, now these neurotransmitters will cross this small space and bind with the receptor, and if it is excitatory, it will cause gates to open to allow sodium to come in to start that depolarization process. So under your notes, number 2, process. Calcium gates open, so here we are right here, action potential first, and then calcium gates open on the presynaptic neuron. Calcium enters by facilitated diffusion. It's using a protein. Why? Because it's got a charge, right? It can't cross this phospholipid bilayer by itself. It needs to use a protein to do that. Calcium fuses with the synaptic vesicles inside the presynaptic neuron, which then migrate and fuse right here to the edge, okay, of the presynaptic membrane. Neurotransmitters, here in purple triangles here, are released from the synaptic vessels into the cleft, this space into the cleft, and diffuse across the cleft, going from high to low concentration. Then the neurotransmitters bind to receptors on the postsynaptic neuron, which can either trigger excitation and the opening of sodium gates trying to reach the threshold, excitation, the opening of sodium gates, or inhibition and a tighter seal on the sodium gates, locking them down, preventing sodium gates from opening up in this area so it does not trigger an action potential. Okay, so remember how we said 1,000 to 10,000 synapses. So you've got to add it all up. That synaptic integration of the postsynaptic neuron. So for instance, this could be an excitatory messages that you're receiving, but you're also receiving all of these inhibitory messages. So when you kind of cancel each other out, overall you have an excitatory message, but not enough to cross the threshold. Okay. And by the way, even if we didn't have this inhibitory message, would we have enough to cross the threshold? No, the excitatory message you had wasn't even enough to trigger an action potential. So integration is the postsynaptic neuron adds up all the excitatory and inhibitory messages from all the synapses and determines whether to send a message as an action potential or not. Okay. Now, these neurotransmitters, you have 100 plus neurotransmitters. Just a couple I want to point out to you that can excite or inhibit. Acetylcholine, known as ACH, acetylcholine. Okay, it can both be excitatory and inhibitory. So it can be excitatory for skeletal muscles. So it's excitatory for skeletal muscles, but it inhibits your cardiac muscle, your heart. Okay, endorphins. All three of these, norepinephrine, think of like noradrenaline, okay, dopamine, serotonin. These are all endorphins, and these are your good feelings, like your runner's high when you're working out, okay? And then dopamine in particular is about emotions and learning and paying attention, which I hope you are doing. Okay, and this is going to conclude video 1, okay? And then in video 2, I'm going to talk to you about the central nervous system. I'll talk to you about the peripheral nervous system and some of the disorders that we have. And if you are one of my students, I'll see you in class.